Okay, so welcome to this next video in which we are discussing the adrenoreceptors. Okay, so we're in the process of discussing the alpha-2 adrenoreceptor, which we know causes presynaptic inhibition. Okay, so what we discussed is that the way it does this is by activating the GI heterotrimeric G protein, but in this case we're not particularly interested in the alpha subunit and what it's going to go on to activate or inactivate. We're more interested in the beta gamma subunit, and I've told you that what it's going to do is it's going to trigger hyperpolarization of the electrical potential difference across the cell membrane, and it's also going to trigger the inactivation of the voltage-gated calcium channels. So I want to discuss the main target for the beta gamma subunit with regards to how it produces hyperpolarization. Okay, so basically it's going to target a type of potassium channel known as the G protein gated potassium channel. Okay, and this is within the family of inwardly rectifying potassium channels. So I'm going to start off with talking about the family of inwardly rectifying potassium channels, and then I'll talk about the G protein gated potassium channel. So inwardly rectifying potassium channels. And inwardly rectifying potassium channels are often abbreviated to KIRs for short. Okay, so inwardly rectifying potassium channels, and they're abbreviated to KIR for short. So K for potassium channel, IR for inwardly rectifying. Right, so basically let's have a look at the structure of these inwardly rectifying potassium channels. So basically, let this be the plasma membrane here, and then I'll draw the inwardly rectifying potassium channel like so. And basically, they are tetramers, okay? So they are made up of 4 separate proteins. So here is the channel through the middle, and then we've got these 4 separate proteins making up the inwardly rectifying potassium channel, like so. Okay, now again, same protocol as for the IP3 receptor. Let's take out one of these subunits of the inwardly rectifying potassium channel, and let's have a look at its membrane spanning topology. So I'm going to pull out this pink subunit of the inwardly rectifying potassium channel, and I want to have a look at its membrane spanning topology. And again, this is the extracellular fluid side of the membrane, and this is the cytoplasm side of the membrane, and it's the same protocol for this piece of membrane that I'm drawing here. So basically what you have is the amino terminus on the intracellular aspect of the membrane, then you have the P-loop, firstly you have a membrane spanning alpha helix, then you have this P-loop which tries to get through the membrane but fails, and then you have the second membrane spanning alpha helix, and then you finish with the carboxylic acid terminus down here. Okay, so this is the membrane spanning topology of a subunit of an inwardly rectifying potassium channel. Now, what would be beautifully simple is if there was just one subunit for an inwardly rectifying potassium channel, and you use this subunit 4 times and you make an inwardly rectifying potassium channel. Okay, so you join 4 of them together to make the whole channel, and that's the inwardly rectifying potassium channel. It's not that simple. Instead, you actually have 15 different genes which all code for different subunits which you can build inwardly rectifying potassium channels out of. Now, these genes are called KIRs for short, okay, for potassium channel subunit that's inwardly rectifying. Okay, so basically to help us understand the inwardly rectifying potassium channels, the inwardly rectifying potassium channel subunits have been grouped into 7 families. So there is the KIR1 family, there is the KIR2 family, there is the KIR3 family, there is the KIR4 family, there is the KIR5 family, there is the KIR6 family, and then finally there is the KIR7 family. Okay, now within the first family you only have one member, okay, which is the KIR1.1 gene, which produces the protein, the KIR 1.1 subunit, which makes up a quarter of a inwardly rectifying potassium channel. Okay, within the KIR2 family, it's a slightly bigger family, you have 4 members. So you have KIR 2.1 all the way down to KIR 2.4. Okay, so you have KIR 2.1, KIR 2.2, KIR 2.3, and then finally KIR 2.4. It's the same within the KIR3 family. You have KIR 3.1, KIR 3.2, KIR 3.3, and then all the way down to KIR 3.4. Okay, then we also have KIR4 family. This contains 2 members. It contains KIR 4.1 and then KIR 4.2. Okay, KIR5 only contains 1 member, the KIR 5.1 subunit. Okay, KIR6 contains 2 members, KIR 6.1 and KIR 6.2. And then finally, KIR7 contains 1 member within that family, which is the KIR 7.1. Okay, right. So that gives us overall 15 different genes and 15 different subunits for inwardly rectifying potassium channels. 1, 5, 9, 10, 11, 12, 13, 14, 15. Okay, so those are the subunits for inwardly rectifying potassium channels. The question then emerges, how do you join these subunits together to make tetramers? Well, basically, you can produce homotetramers, but you can also produce heterotetramers. So homotetramer is basically where you use the same KIR subunit in all 4 slots. Okay, so you make a inwardly rectifying potassium channel out of 4 identical inwardly rectifying potassium channel subunits. Okay, and that will be called a homotetramer. Homo for same, tetramer for 4-membered structure. Okay, so it's made up of 4 of the same subunits, basically. However, you can also produce heterotetramers, and hetero obviously means different, so this means that the tetramer will be made out of 4 subunits which are not all the same, basically. Okay, now you might be wondering, well, what's the scope with that? I.e., can I just pick 4 subunits of my choice? You know, I just pick 4 of them and stick them together and make a inwardly rectifying potassium channel? Well, the answer is no. You don't really find heterotetramers quite like that in nature. The rules that we see for nature seem to conform to this. Okay, so I'll tell you the rules by which nature seems to build inwardly rectifying potassium channels. The rule seems to be that you firstly choose a family. Okay, so you choose one of the 7 families. Okay, so maybe I'll choose the KIR3 family. Okay, and this is why the naming was created, to try and produce a nice system by which we can understand how to build heterotetramers. So you first choose one of the families, and in this family, what you then do is you pick up to 2 separate subunits that are within this same family. Okay, so you could just produce pick one rather. If you pick 1, then you can make the homotetramer for that. And some of the homotetramers are found. For instance, the ones that are in a family on their own, such as KIR1.1, which is in this KIR1 family on its own, it will produce a homotetramer where the 4 subunits are all KIR1.1s. But if you're in a bigger family like this, generally you don't just pick 1 of the members of this family. Instead, you will pick 2 of them, and then you can produce heterotetramers which contain those 2, basically. So, for instance, you can pick KIR 3.1 and KIR 3.4, okay, and you could produce a heterotetramer which contains both KIR 3.1 proteins and KIR 3.4 proteins. Okay, the next question you might have is then, what's the stoichiometry of them? I.e., Do I have one KIR 3.1 and 3 KIR 3.4s, or do I have two of both of them? And then once I've decided on how many of each I'm having, how do I decide how to orient them relative to one another? Okay, those sort of questions are not known, basically. We don't know what the stoichiometries of the heterotetramers are generally. We don't know what the orientation of them are. Okay, what we do know is that the heterotetramers only generally contain two different KIR subunits, and the two different KIR subunits are generally from the same family, which is why we've created this naming system, basically. Occasionally there are breakings in the rules, okay, so there are some heterotetramers where you've got mixing between the families, but we haven't found any breaking of the rule that you only use two different KIR subunits, okay, it's just the naming system isn't perfect, basically. Okay, right, so basically, these are the official names, but there are also kind of colloquial names for inwardly rectifying potassium channels, which are built out of subunits from each of these families. Okay, so for instance, the inwardly rectifying potassium channels, which are built out of subunits from the KIR3 family, these are the G protein gated potassium channels. So this is actually the family of inwardly rectifying potassium channels subunits that we are interested in. So this is the G protein gated potassium channels. So if you produce me an inwardly rectifying potassium channel which is produced out of subunits which are within the KIR3 family, then that inwardly rectifying potassium channel will be called a G protein gated potassium channel. Okay. If on the other hand you produce me an inwardly rectifying potassium channel where the subunits of that inwardly rectifying potassium channel are from the KIR2 family, then those inwardly rectifying potassium channels will be called classical inwardly rectifying potassium channels. So where should I write this? So these are the classical inwardly rectifying potassium channels. Okay, so I'll just put KIR channels. So classical inwardly rectifying potassium channels. Okay, if you produce me an inwardly rectifying potassium channel which is built out of subunits from the KIR6 family, then again this is a very important family. Okay, not for this video, but a very important family in pharmacology. These are what are known as the ATP-sensitive potassium channels. Okay, so these are ATP-sensitive potassium channels. Okay, and finally, if you produce inwardly rectifying potassium channels where the subunits of the inwardly rectifying potassium channels are from any of the other remaining four families, so this one, this one, this one, and this one, then you will be known as a potassium transport channel. Okay, so all the other families, the potassium channels that you can produce from those are called potassium transport channels. Now for us, we're going to be extremely interested in these G protein gated potassium channels, and we've gone to this discussion of the inwardly rectifying potassium channels so that you actually know where these things come from. They are part of this larger family of inwardly rectifying potassium channels. Okay, so we're going to be interested in homotetramers and heterotetramers produced by subunits within this third family. Now, you don't generally produce homotetramers of this family. Instead, there are three main heterotetramers which are found in nature. Okay, so the three main heterotetramers which you find within nature are the KIR 3.1 with the KIR 3.2 subunit, the KIR 3.1 with the KIR 3.3 subunit, and then also the KIR 3.1 with the KIR 3.4 subunit. So you can see from that that it's the KIR 3.1 subunit of the inwardly rectifying potassium channel paired with every other option it has, basically 3.2, 3.3 and 3.4. So these are the three heterotetramers of the inwardly rectifying potassium channels subunits that you generally actually find producing these G protein gated potassium channels. Okay, right. So, what's going to happen then now? Basically, these channels are not usually open, okay? But the beta-gamma subunit of the heterotrimeric G protein can come and bind to these channels. So here it comes. Here's the beta-gamma subunit, and it's in the phospholipid there, and it's going to come along to one of these G protein gated potassium channels over here, and basically it can bind to it, and what it will trigger is the opening of this G protein gated potassium channel. Okay, now what happens if the G protein gated potassium channel opens? Well, we know what the normal electrical potential difference across the membrane is. We know that it's usually around -65, which means that the intracellular electrical potential is around 65 millivolts lower than the extracellular electrical potential. Okay, so this is usually around -65 millivolts. Now, we also know that potassium concentration inside the cell is around 155 millimolar, and extracellularly it's around 4 millimolar. So, here's a conundrum. The potassium concentration gradient says move potassium out. The electrical potential gradient, on the other hand, says the electrical potential intracellularly is actually 65 millivolts lower than the electrical potential extracellularly. Potassium is a positively charged ion, so it will prefer to be in the intracellular compartment, which has the lower electrical potential, lower by 65 millivolts. So, the electrical gradient says go in, the concentration gradient says go out. Which one is stronger? Well, I've actually already given you the answer to this. I told you earlier that the Nernst potential for potassium at the normal concentrations across the cell membrane is around -85 millivolts. What that means is that actually, to completely oppose the concentration gradient, the electrical gradient across the membrane needed would be -85 millivolts, i.e. if the electrical potential of the intracellular compartment was 85 millivolts lower than the electrical potential of the extracellular compartment, then that would be strong enough to stop the movement of potassium ions out of the cell because of the concentration gradient. If you went beyond that, if you say went to -95 millivolts, then you would actually start getting potassium ions moving back in, because the electrical gradient would be bigger than the concentration gradient. But at -65 millivolts, the electrical gradient is too small to counter the concentration gradient. So it will reduce the movement of potassium ions out, certainly from if the electrical potential difference was neutral, but it won't completely oppose it. So you'll still get a small net movement of potassium ions out, shown here in green, which is just the pink arrow added to the blue arrow. Okay, now what does this trigger? Well, we are moving positively charged ions from the intracellular compartment to the extracellular compartment. So we're raising the electrical potential of the extracellular compartment by bringing positive charge in to the extracellular compartment, and we're reducing the electrical potential of the intracellular compartment by removing positive charge from the intracellular compartment. So, if we now ask how much lower is the intracellular electrical potential than the extracellular electrical potential, the answer is the number's going to get bigger, basically, because we're raising this number and lowering this number. So the amount will go down when we move from here to here is going to get bigger. So this number is going to become more negative, i.e. it's going to become hyperpolarized. So this then is how the beta-gamma subunit of the heterotrimeric G protein is capable of hyperpolarizing the membrane of the axon terminal of the neuron, and that is going to stop action potentials from being able to propagate into the axon terminal as easily. Okay, right, so we'll call it there for this video, and we'll discuss voltage-gated calcium channels and the action of the beta-gamma subunit on voltage-gated calcium channels in the next video.